Go ahead, Ben.
Okay, great. So welcome Jack Stapleton, who is an infectious disease physician from the University of Iowa, who's been doing some very interesting research on GBVC for the human pegivirus over the past decades, really now, I guess. So so we'll keep going with our conversation, but Jack, so the pegivirus is P for persistent and G reflecting the historical name of the virus is pretty it's a pretty amazing story and a pretty amazing, I guess, discovery story. Given that in the mid-90s, two separate groups of researchers identified a new virus using serum from the 60s from a marmoset, which was injected from with blood from a surgeon whose initials were GB in an experimental procedure. Um, can you explain a little bit? But of course, the name is was called GBVC, now called human pegivirus. Can you explain a little bit, perhaps, the kind of
Sure,
the move from GBVC
sure,
to pegivirus?
sure. And actually, the two different groups discovered it using different samples, but in the mid 90s, it was clear after Hep C was identified that there was a large group of people who developed hepatitis post transfusion who didn't have A, B, or C.
Mm,
So people were looking for other agents that could cause hepatitis, transfusion related hepatitis. And so Abbott Laboratories had with Fritz Dienhart at Rush University had in the early 60s had a surgeon whose initials were GB who developed fulminant hepatitis. And Fritz put these into marmosets and they got hepatitis and he was able to passage it. So it was clearly an infectious agent. And in the 12th passage of the marmosets, they made a pool of virus and tissues, and a lot of people, Steve Feinstein at NIH and lots of different people use those tissues to look for hepatitis A and B. And the GB viruses never really panned out as tissues, never had any relationship to A or B. And in fact, we're part of the NIH blinded serum pool that was used to test for putative Hep C agents. And when Chiron discovered Hep C, the only one in the panel that didn't react with their Hep C antigens and was GB virus. So it was interesting that GB didn't have Hep C, and in retrospect, the surgeon didn't have human GB virus
mm,
either. So he did and then the other group looking was Gene Labs, and they were using tissues from people who had non-A, non-B, non-C hepatitis and using subtraction methods to subtract out normal sequences. And they, using those tissues, found what they called hepatitis G.
mm,
But once they started looking, it was really everywhere, and people there was no association with acute or chronic hepatitis, and so it was because it's transmitted similarly to Hep C and HIV. It turned out the prevalences were much higher in people with those transmission
mm,
exposures, but it wasn't by itself associated with any disease.
mm,
So because of that, because it didn't cause hepatitis,
mm,
hepatitis G was not a great name.
mm,
And because GB, the surgeon, didn't have it, and it turns out the marmosets that got hepatitis had two GB agents that were discovered before human GB.
mm,
And it turns out that one of those, GBVB, was what causes hepatitis in marmosets. So it was actually one of the viruses that was in the captive animals that was
mm,
causing the hepatitis. And it just happened they injected the human serum into one of them that you know, uh,
mm,
just a good circumstance that happened, you know, an opportunistic circumstance that led them to believe that. So because GB didn't have it, didn't cause hepatitis, I and four colleagues of mine, one at Abbott, one at Gene Labs, Jens Buch in Sweden, and Peter Simmons in Edinburgh at the time. He's at Oxford now, actually. Peter is. Um, we thought it was a bad name, and so we wrote a review article and summarized all the features, why it was deserved to be called be part of a new genus. And within the Flaviviridae family. And so we named the genus Pegi for persistent, because the GB virus and Hep G were capable of causing long-term what are persistent infection in humans, which most RNA viruses don't. So it's persistent, and then G was just for the kind of the nod to the historical discovery by the two groups. So that's how it became a pegivirus.
Mm. Yeah, thank you. And I should say that feel free to jump in, Evan or Jonathan, if you've got any question. Or I can see Nunki as well, who's an Indonesian colleague. I don't know if you've met Nunki before, Jack, but
I don't believe so.
Yeah, did her PhD with a researcher in Kobe. I published a paper on pegivirus in Indonesia. But yeah, I mean, so GB, I know that you've published an article that kind of speculated whether GB should be not stand for the name of the surgeon, the initials of the surgeon, but for good boy. Good boy referring to the non-pathogenic kind of, or symbiotic kind of, apparently symbiotic role that the virus plays. I'm interested in a way in, so I see a series of debates emerge in New England Journal of Medicine in the mid 2000s, which kind of discuss the role of human pegivirus in cases of co-infection with HIV. And you kind of conclude in your review essay that HBGV, which is another name for human pegivirus, appears to represent a largely symbiotic infection within the human virome, little to no pathogenicity. So can you tell us a little bit more about how you came to be interested in GB virus? I mean, I think HIV is clearly a part of the story, but yeah, and a potential beneficial role.
Like many things in science, it was serendipitous.
Yeah.
I was a clinically, I was hired by Iowa to set up their HIV program. I was a pretty rural state and didn't have much HIV, and they were just starting to have cases. And I trained in UNC with Mike Cohen and people like that, and had quite a bit of experience with HIV early in the 80s. So, although our AIDS. And so I set up a clinic here, and as part of that, I set it up so that I would have collect serum on everybody when they came in as a potential research tool, and set up a database. And my lab was hepatitis A vaccine related, and so from when I got here in '86 until '94 or '5, my funding was in the lab was all pretty much for hepatitis A virus, antigenic structure, immunogenicity type work, and I learned early in the course of things that if you aren't willing to change and expand, you can not have funding, because I had a lot of great data in '93, and my main source of funding when I went for my competitive renewal wasn't renewed, because who needs to do Hep A research when there's a good vaccine? Mm-hmm. And so I rewrote and resubmitted. Back then you got 3 shots, and between the second and third one, I pretty much quit working on Hep A, because I'd already done a lot of the stuff I'd proposed by then. And lo and behold, I got the Hep A grant, but I'd already done most of the work. So I had 4 years to work on new things, and I started working in '93 on this new hepatitis virus, hepatitis C virus. And then I also started set up some HIV lab systems so that we could do some kind of supportive virology for some of our clinical trials work we were doing. So when GBVC was discovered in GBV A and B were discovered in '95, and GBVC in '96. I had a lot of samples in the freezer and things like that from hepatitis patients and from HIV patients. And like a lot of labs, we pulled everything out of our freezers and tested to see if GBVC was there. And I got interested in because it's a close relative of Hep C, on some of the things we're working on with Hep C, like cell binding and entry, and the core protein, since the particle doesn't seem to have a core protein. And so we started working on that as kind of side projects for Hep C projects. And at the 2000 Hep C meeting in Bethesda, there were 2 groups, one in Germany and one in Japan, They had pretty small studies, but they were looking to see if GBVC caused hepatitis in patients living with HIV, and it didn't, as what we had found. But when they looked at survival, they said it looks like people live longer if they have this virus. I remember Donald Smith, who works with Peter Simmons at the time, running into him for a beer, and he said, "What do you think of that?" And I said, "It's probably a bunch of crap, but we're gonna find out, because I've got samples on everybody who walked in the door in our clinic, and we'll go home and test them and see." And to be honest, I was pretty shocked that patients who were G positive when they walked in the door were 3 times more likely to be alive in follow-up, up to 10 years of follow-up. And this is back in the late 80s, early 90s, when we really didn't have effective therapy. So it was pretty shocking, but it led to the opportunity to spend 25 years trying to figure out why it might do that.
Yeah, amazing.
So circuitous and not planned.
Yeah.
So I've actually got a question that follows on from that, and I've read the 3 papers, or at least skimmed them, that you've offered us tonight, and tonight Oxford time, today your time, yeah. So I guess I'm just wondering if this association with positive survival rates in HIV is an accident of history, or do you think there's something specific about this virus and HIV virus? Because some of these newer papers seem to suggest that it might be quite broad effects that pegivirus has.
Yeah, no, I don't think it evolved these mechanisms for HIV. I think the way I look at it, I mean, we went to with what viral proteins are specific inhibitors, and we looked at NS5A and E2 and found those 2 specifically inhibit, whereas others didn't seem to in our systems we were looking at. And the virus did inhibit, but it took us a while. It took us probably 10 years to really come to the conclusion that it's really, while there are specific mechanisms that inhibit HIV, and the main effect seems to be on reducing T cell activation. And the fact that this virus can replicate at 50 to 500 million copies per mil in plasma, and you have reduced activation, T cell activation, B cell activation, monocyte activation, and you don't make antibodies in a consistent way to any of the viral proteins, is pretty remarkable. And so I still think that that's a really an amazing feature of this, and I think it's evolved with humans as an anti-inflammatory that doesn't knock out your immune system, but it dampens it as a rheostat, so that it protects you against immune-mediated diseases. We did one study in lupus patients to see if it was associated with a reduced prevalence of lupus, and we found no infection in 100 and some patients with lupus, and we found and there was a significant difference between our healthy control, but it was only like 3 in that, or 5 or something. The problem is it's very low prevalence, and you need large numbers. And I applied a few grants to try and get funding to do that. Nobody was very interested in it, but I think many studies of diseases that are immune mediated, very few, if any, take into account GBVC as a part of the virome. And I think they should, especially in populations where there's a lot of virus, like Hep C, HIV. A quarter of Hep C patients have it. 15 to 40%, depending on the study, have GBVC in HIV populations. And in developing parts of the world, it's high. It's 15 to 30% in regions in Africa, and 5 to 15% in Sweden. And so if that's influencing your activation and you don't account for it in these studies, you're really missing a potentially important variable that may confound your results and actually explain your results sometimes. And it's been really hard to get people to think in those terms. I've about got a guy in the ACTG convinced that we should look at cardiac disease and HIV patients, or people participating in HIV studies. And so we're going to be screening some people looking for that. But Dave O'Connor did the study looking at the sequences. They did deep sequencing of people with Ebola during the outbreak as 2015, I think. And he found that if you had GBVC, you were 4 times less likely to die than if you didn't have GBVC with Ebola infection. So I think a lot, if you hadn't looked, you'd never know those differences. And I do think it's an important part of the virome. And it's complicated in that people clear it spontaneously, and immune-competent people clear it pretty regularly. And you can get reinfected. We don't know any of the rates or factors associated with those reinfection, but it clearly happens. And then it can persist. And what are the host factors that lead to persistence? Those themselves may be variables that also contribute to a dampened immune response. So, it's very complicated epidemiologically, but I'm fully convinced that it's an important actor in human disease, many diseases.
Yeah.
Super interesting.
I'm enthusiastic about it. I don't know if you all saw the group in Sweden or Switzerland looked at stem cell transplant recipients and found a reduction in graft versus host disease and improvement in all-cause survival. Now, it's a small study, and it's hard because there are a lot of other viruses in there, and the timing when you do it, it can come up and down, and so it's difficult. But it is, I think, a fascinating story.
I've got one question that's just kind of like maybe a question about stigma. I mean, do you think it's, I think there's been a gradual acceptance of bacteria as, something that can be other than pathogenic. There's been a boom even of both popular writing and I think scientific work on the microbiome. But do you think there's some kind of special stigma that is attached to viruses that prevents people, even scientists, from fully appreciating that, an infectious agent like this can be something other than pathogenic?
I don't know if it's that. I think there's a degree of skepticism that's always good in science, and there aren't. I think it's fascinating that with deep sequencing we haven't found more bloodborne viruses in the virome. That we aren't finding new viruses right and left. We are, if you look at animals, we're finding all these viruses that you can't cultivate that we thought we didn't know of. There are about 90 different hep C and pegiviruses now with different hosts. And 10 years ago there were like 4 or 5. So I think deep sequencing has turned up new infections of these viruses in different species, but it's not turning up new viruses right and left in humans or in other animals. And so I think there's a much more limited bloodborne virome than of course you don't find bacteria in the bloodstream either. And then because they're intracellular pathogens, and I don't know, in the GI tract, there are a lot of viruses there. I haven't really pursued those to see what they do to the I don't think there's a lot known on those, to be honest. But so I think there are Skip Virgin published a paper in Science not long after we published some of our early work on GBVC and HIV, and showed that you could, in an animal model, use a murine herpes virus that with a single mutation you prevent latency. And so he did a latent herpes, I think it's 68 or something, I don't remember the number for sure, but it's a murine herpes virus. Infections of mice with the persistent infection or the latent infection or not, and showed that the mice who got the same virus but didn't develop latency were actually more susceptible to death by listeria, by secondary superinfection with bacteria. And he attributed it to, he showed that it was related to interferon by using neutralizing antibodies and things like that. And I remember Phil Murphy sent me an email and said, this got into science. You already showed this like 5 years ago, that there's this competition between viruses and the effects on the immune system. But so I think those things do exist, and we don't understand them well. A guy at NIH named Leonid Margolis has done a lot of work on herpes virus infections and HIV, and there are a lot of effects of these long-standing viruses in humans that we don't understand, that do influence disease outcomes and so forth. So I think it's real interesting. But why people don't appreciate that is that I think they probably will. And if you ask them, most people would probably say, yeah, I suppose they could. We just don't know much about it, maybe.
Yeah. I mean, I do see some, I mean, the other infection, of course, is coronavirus. So I don't know whether you've seen any studies, Jack, related to co-infection with coronavirus.
I really wish we would. We have a fairly small lab and we just don't have time, but I'd love to look at severe disease and not severe disease. And I've talked to several people who say, oh yeah, we can do that. And then kind of it's petered out. We haven't wrote down, but I would think that since the severe disease is immunologically mediated, that it would not surprise me if it's ameliorated somewhat in people with GVC compared to not. But then people say, so what? What are you going to do about it? So funding has been tough. The few labs that have gotten into this have struggled to maintain funding.
Yeah, interesting. I mean, there is this, emerging interest, and Eben, of course, can speak more to, of course, in symbiotic, virus and non-pathogenic role of viruses. And in Japan, I mean, I was looking up the other day. In Japan, there's this kind of neovirology as a kind of field, and quite a large group is looking at precisely the kinds of questions, I guess, that you're asking, Jack. So potentially there's a moment at which this research is going to be, playing a more prominent role than it has otherwise.
Well, one of the reasons GVC studies have gone so slowly and it's still really difficult because we can't culture it well. It's like Hep C research, and Hep C research had lots of big labs around the world working on it for a couple decades before really you were able to grow the virus. And then it took a very select isolate and cells that had been selected for adaptation to the replicons and things like that to get a system where you could grow the virus in vitro. If we could grow GBVC, I think it would be an amazing gene therapy vector. And I've written a couple grants on that. I even got one funded early on in the 90s to try and set up a cell culture system. But I and David Thomas at Hopkins and Rajal Bhattarai at FDA and lots of different people have tried to either use a macaque model to have an animal model for a human virus, or to set up lots of different cell culture approaches have been attempted, and so far nobody's gotten it to work. The closest thing has been Dave O'Connor at Wisconsin got a baboon virus to grow in macaques, although he wasn't able to get human virus to grow. So it's very species specific.
Do you have a sense of like what receptors it's using to get in the cell? Are these like species specific receptors, or? So
there's one paper suggesting that the LDL receptor is involved from Vince Agnello from back in around 2000. And I think it probably can, because the particles appear to be lipid coated. And so I think it probably can't, like Hep C, particles in serum can probably bind the LDL receptor and get in, which go into every cell. But I don't, my thinking process is that it utilizes a receptor that's transiently expressed in stem cells, because when you look in cadaver studies from the 90s to David's work in primates to basically everywhere where you look, you find virus predominantly produced in the bone marrow. And so I suspect it's a stem cell that has a very transiently expressed receptor that gets in and makes a lot of virus. And as those cells differentiate, they lose that receptor, and therefore the virus doesn't kill them. So you get virus released in all kinds of cells, and even maybe some low-level production, but not enough to really account for being the primary target of the virus. And we did these studies, Ernest Chavera, when he was in my lab, did these studies where we purified lots of different types of T cells, monocytes, NK cells in peripheral blood, and we found virus in all of them, basically. And a couple of people have published that you find virus in platelets. It's interesting, if you take PBMCs out of a patient with HIV who's co-infected and just put them into cells, into media, and keep them at a good temperature, about most of the cells die in the first 1 week, 7 days, or 7 to 10 days, like you'd expect. And then there's a small population of cells that just sits there and doesn't do anything. And Ernest kept them going for over 80 days. And they release very low levels of virus in the supernatant. We think it's because the way GBVC inhibits lymphocyte proliferation is it competitively competes with the lymphocyte specific Src kinase, which is required for proliferation. And so that by doing that, it really makes those cells not grow. It also makes it hard to grow then in those. If you have cells where the virus slows down the growth of the cells that in a transformed cell culture model, those other cells that are not infected will outgrow and will get diluted out. So you lose the few infected cells you have, unless you're lucky and get the right type of receptor and have it expressed over a long period of time. And nobody's been able to do that yet.
Jonathan, I can see you've come on camera. Did you have a question? Sorry, it's Jonathan, isn't it?
Oh, thanks. I was just joining, but I did have something on my mind. Did you say, it's possible I misheard, but did you say that this virus also was associated with improved outcomes in Ebola patients?
Yeah, there's one paper that suggests that. It's Dave O'Connor got the sequences that were published when they did the GWAS stuff on the Ebola, people with Ebola from West Africa outbreak. In 2015, there was a paper describing all the genetics in science, and he knew the woman, and he called her and said, "Can I get you the sequences and we'll look for GBVC?" And he found it in quite a high percentage of those people, and in those who he published it in J Virology, which I thought was a mistake. He should have gone to somewhere where people interested in clinical outcomes would be see. But anyway, the mortality rate was like 80% in those without GBVC and 20% in those with GBVC.
Wow.
I wanted to ask about that, that when you were mentioning the outcomes with HIV patients and the work that you wanted to do on lupus, it sounded like the mechanism was, I guess, dampening the body's immune response so that autoimmune effects or immune deficiency would be reduced. But it seems like with Ebola, it would have to be a different mechanism, right?
I don't think so, because I mean, not only do you have incredibly high levels of virus, but you have cytokine storm type effect too, right? Okay. So it's and there are a lot of animal models, like with Coxsackie virus. Coxsackie virus cardiomyopathy, if you treat mice, you put them on a treadmill. There's all these exercise studies from the 60s and 70s. Infect them with Coxsackie. If you treat them with steroids, they don't die as much, and they don't get as much cardiomyopathy, but they have more virus in their heart. So it's a balance, I think, between the direct effects of the virus and the immune effects of the virus. And I think with Ebola, what kills people is more the immune effects. Like in SARS-CoV-2, I would not be surprised, but I don't know if that's true for that as well.
Thank you.
That's my opinion. It's speculation.
I appreciate it.
And maybe to just summarize, so the speculation is that it reduces cytokine storms and
I'd even make it more general. I'd say it reduces inflammation.
okay.
So it's an anti-inflammatory virus.
So there's probably some conditions where it wouldn't be good to have that anti-inflammatory effects.
Yeah. But it doesn't shut them, it doesn't like ablate those inflammation. So when, we've got all these studies where we show T cells when you incubate them in GBVC and then you stimulate them through the T cell receptor, you see this, you see like a level this high for the controls and this high for the GBVC incubated versus none for the non-stimulated control. So it doesn't prevent appropriate responses by the cells. It just requires a bigger hammer to bigger stimulus to stimulate a lower response. And so in an infection, if most infections or a lot of infections are really mediated by tissue damage from inflammation, from CD8s going in and killing infected cells and attracting other immune cells that are going to destroy local tissues, and so I think by dampening that but not getting rid of it, it's beneficial. And I think that's why it stayed in humans since Lucy. Again, pure speculation, but
actually, has there been any ancient DNA work, or is it, actually I don't even know if it's a DNA or RNA virus showing
RNA, it's an RNA virus. That makes it tough.
Okay.
I think Peter Simmons, actually, you're at Oxford, and Donald Smith, they published a lot of this, and I stole a lot of it, and I always credit them. But I mean, the lines of evidence are pretty interesting that it is an ancient virus, because if you go to indigenous populations, and 4 or 5 studies now have been done where you look at indigenous populations of peoples who are not intermarried with non-indigenous peoples in South America and Papua New Guinea and things, and they all have the Southern Asia, Eastern Asia genotype. And then you go into, say, Medellin, Colombia, and they have a mixture of genotype 2 and 3. And genotype 2 is the one in Western Europe and North America. And so there's a lot of segregation in peoples that supports that it's been in indigenous peoples since they got to South America, for example. And I like the part that if you look at the genotype, the differences in genetic genotype or DNA, or the phylogeny of GBVC, the 5 genotypes share the differences. Genetic distance between genotypes is the same as the genetic distance between mitochondrial DNA and human races. That was Peter's on his studies. So it suggests that it entered humans before and then the geographic distribution of the genotypes is very much human migration out of Africa, distribution of genotypes, predominant genotypes. And then if you look in old world and new world primates on both continents, the old world primates are on both, and the new world are only in the new world, pretty much. So it suggests that my understanding is that suggests that they were in primates before the continent split, but I don't know if that's true or not. I could be making that up. It sounds cool. I don't know if it's real. I have no expertise in that. But so I think there's several lines of evidence that suggest that it isn't really an ancient virus, and there's no way RNA is too unstable, so you'll never get RNA from ancient samples.
Hmm.
I suppose. I don't know if it, the Jurassic Park stuff with the
I guess that like the recent some of the work that you cover in your article, Jack, you're speaking about the wide distribution across species as well, and of course so when the virus, although it was the GB serum, it was actually it was the marmoset, I believe that
yes.
was the host, yeah, for this.
The Abbott folks never found the human virus in the marmosets, but they used degenerate primers from the marmoset viruses to screen humans, and then they found some, and then they made better primers, and they found that it was really common.
Yeah. So the virus in the marmoset is quite similar to the human pegivirus, is that?
Yeah, and they're pretty high. They're very highly conserved. So the 5 genotypes have less diversity than a subtype of a genotype of Hep C, for example. There's far more diversity in Hep C.
Mm-hmm.
And this goose, the goose pegivirus is really interesting. So it's in avian species also. It's in geckos. It's in sharks. They're finding it everywhere they look. And the geese, they were able, it hadn't been replicated yet, but it's really interesting. They've been able to get the virus to grow in embryonated eggs. So that may be a culture system. And again, would suggest that you need a stem cell type of environment for the virus to grow.
So it's the embryonated goose eggs that might provide the.
Well, for goose pegi, but not for human.
Yeah.
And there's some major differences within the pegiviruses between species. There's an X protein that's different, and the size of some of the proteins are quite different, but the overall structure is otherwise similar. There's homology between the polymerase and the NS5A and the protease regions.
Great. And so I mean, just looking back at the kind of, I guess, research into the pegivirus, you can see waves of interest at points where it's kind of correlated with pathogenic, there's a potential pathogenic cause. So of course, hepatitis C being the kind of intensive period of research. Recently I've seen, well, there's some studies that suggest lymphoma, that human pegivirus causes, may play a role in causing increased infection with pegivirus is associated with higher risk of lymphoma. What's your kind of understanding of that?
Yeah, so I'm actually on 2 or 3 of those papers, 4 or 5 of those papers. And I think it's real. I think there is, in people who are persistently infected, they have an increased risk of non-Hodgkin lymphoma by about 2-fold. So you're talking about 1 in 4,000 people instead of 1 in 8,000 people get non-Hodgkin or get lymphoma of some type. So it's not it's a doubling, which is significant, but it's not a huge impact on human health. But my personal take is that it's not a direct effect of the virus, that it's a dampening of immune surveillance. Because we all make neoplastic lymphocytes and they get cleared by our immune system. And if you have a slightly weakened recognition system because your antigen presenting cells aren't quite as turned on as they could be, maybe you have more escapes, but that's also pure speculation. The other one that's very interesting is Chris Powers' stuff from Canada, and I helped him with some of the lab work he's done. He's found pegivirus in cases of sporadic encephalitis, and he's found it in brain, and he's shown that he's been able to show viral antigens in some of the cells in the brain and the histopathology, and he's trying to do work on that. And so I think if that's true, it must be a host factor, because if you've got 3% of healthy blood donors and and a third of the HIV patients and a third and a quarter of the Hep C patients, you'd expect a lot more to have encephalitis if it were direct effect, but it may be that there's a host defect in some people that predisposes to that response to PAGI, but it may well be in some of the CNS tissues. So I think that's an interesting finding. It's just in the last 3 or 4 years that that's come about. And he's working very actively on that. I still have meetings with him once in a while. But so I think that's the other place where there might be pathogenicity. When we had our first study of our clinic, I got invited to Fauci's lab to give their seminar in 2001, and the comment was, "What you found is really interesting, but you really don't know how long they've been infected with HIV, and there's a lot of variables," which I totally agree with, even though we controlled for what we could. And they said, if you find that in our max cohort, it'll be important. So we collaborated and did a study in seroconverters in the max, and that's a 2004 paper with Carolyn Williams, was first author. And in seroconverters, it was pretty interesting that in the short term, it didn't affect survival. But if you looked longer term, over the first 5 years, you didn't really see a difference in mortality. It took a longer period of time, and the immune activation that drives HIV pathogenesis does take time in a lot of people. So I think that fits. The other thing was a fifth of the people who tested during the early part of HIV cleared their GBVC viremia, and if you cleared viremia, you were more likely to die than if you didn't. And that was true in a couple other studies as well. And then a guy named Greg Bassan looked at seroconverters in the HPTN network. And what he found was that about a third of pegivirus infections in that cohort acquired pegivirus at the same time they acquired HIV, because we did pre-HIV and post, whereas Max only let us do post-HIV, but in the first year. So I think a lot of people who acquire HIV will, because there are a lot of people carrying both viruses, will then acquire both. And then if they don't clear, they get a beneficial effect. But if they clear, and whether that's some innate host factor associated with clearance of virus that's bad for you if you have HIV or what, we don't know.
I was wondering if you could speculate a little bit more about just pathways of transmission. So you've talked about blood transfusion, and what I was hearing just then is, this is also likely an STI.
Oh, it is it's sexually transmitted.
It is an STI, yeah.
And maternal fetal.
And what are some of the other ways that this virus is getting around?
Whether or not there's any horizontal infection in kids, probably not a lot. If you look at the age of acquisition, the prevalence jumps up in the teens when people are developing sexual maturity. And then people who have just bloodborne exposures are high risk. People who have lots of sex are high risk. So like in the max cohort, the average number of sexual partners in that cohort was over 1,000. And the prevalence was 39% in that group. So it's very high if you're having a lot of sex or a lot of exposure to blood. Maternal fetal varies, but most studies say it's about 15%. And there was a really nice study by Wendy Supaful and CDC from Thailand, showing that if the baby acquired GBVC, there's an 87% reduction in transmission of HIV in those people. And so I think it's really interesting, but people's response kind of is, yeah, but what are you going to do about it? So I think that's kind of short-sighted, and that ultimately we might learn something about the immune system that would help with HIV and other immune-mediated diseases. But until we get a cell culture system, a lot of labs have started, and then when they find out how hard it is to work with, they kind of drop it because they don't get results very fast. Having worked with hepatitis A, which takes 2 to 3 weeks to grow, starting in that, I thought it was fine. And hep C, I always worked with things that don't grow well.
Yeah. Amazing. I mean, the recent kind of, so, 2019, 2020, I think it was in the American Journal of Epidemiology, there was kind of a kind of debate around, the use of human pegivirus as a biovaccine of some
Biotherapy,
description.
yeah.
Yeah, and particularly in developing country contexts or low or resource poor settings, and also settings where you've got high rates of treatment resistant HIV. So I mean, I'm not sure if you followed that, and perhaps we've almost reached an hour, but if you've got any thoughts on the kind of what the future of pegivirus.
I think it's interesting, and I think the idea of using it as a therapy is interesting, but it doesn't prevent HIV infection clearly, because 40% of the max cohort had both. And so I found that paper to be interesting and an interesting way of thinking about it, but I'm not sure how feasible that would be, because people clear it. They didn't take into account the clearance rates very well, and what the reinfection rate is if you re-challenge someone who's cleared, we don't know, but it's reduced. We know that there's some reduction in infection in people with antibodies who get exposed to virus through the liver transplant data. So I think it's I think it's not unreasonable to think about, but I'm not sure how feasible that is. This is a quick answer at the end of the hour.
Great. Well, if there are, Evan, did you have any other, Jonathan?
I just had to say that I've been really enjoying this conversation, Jack. Thank you so much for entering into conversation with us. And I've got to go right at the hour because I have another call. But yeah, grateful for this dialogue and can't wait to learn more.
Well, it's an interesting virus, I think, and having spent almost 30 years working on it, it's sad that I don't have funding, so I do all the projects we do now are on the side.
Yeah, amazing. Thank you so much, Jack, for sharing your time
Nice to
with
meet
us.
you all, and
really appreciate it.
happy to answer emails if questions come up.
Yeah,
Great.
brilliant.
Thanks so much.
Have a nice
Take care. Good to see
day
you all.
wherever you are. Bye.
Yeah, right. Take care.
See you, Evan.